Hey everyone, welcome back. So we are going to move on to the next section here, and really this is just going to be an extension of the last thing that we talked about in the previous video, talking about transmembrane domains and how they mostly are going to consist of hydrophobic side chains of amino acids. So let's talk a little bit more about that here. So if you think about what we've talked about before with how side chains like to interact with each other and with their environment, it should make a lot of sense that a transmembrane domain, again meaning the stretch of amino acids within a membrane protein that actually interacts with the hydrophobic core of the membrane, it should make a lot of sense that this should be mostly made up of hydrophobic side chains. But that's not to say that this domain can just take on any sort of structure that it wants and still be stable. So there's one other thing that we need to consider here. It's not always just about the side chains. We need to think about the backbone as well. And the backbone still contains atoms that are capable of participating in hydrogen bonding, meaning that they're actually polar. We're talking about the nitrogen and the carbonyl oxygen that make up the backbone and make up the peptide bond. So those are still fairly polar atoms. So what that means for us is we can't just have the hydrophobic transmembrane domain just stretched out like it would be in the primary unfolded structure of the protein, because then those polar atoms are fully exposed, and that's not going to make the fatty acid tails in the hydrophobic core of the bilayer very happy. So we need to structure these transmembrane domains in such a way that we try to hide those polar atoms away from the hydrophobic core. The same way that when a soluble protein folds, we try to hide those hydrophobic side chains at the core of the protein. Really the same principle at play here. So the optimal way to structure a transmembrane domain is as an alpha helix. That's one of the most common types of secondary structures that we've had a chance to talk about this semester. So if we form an alpha helix with our transmembrane domain, we should be able to work something out in which the backbone oxygen and nitrogen atoms that we were talking about, they can participate in hydrogen bonds with each other, and they end up buried in the center of the winding helical structure of the alpha helix, and they are hidden away from those phospholipid tails. Again, everyone seems to be happy here, so that always seems like the optimal way to do business, especially if you're a protein. Now, the particular protein that the Lotus book is using as an example, and you can actually see it illustrated here, is a membrane glycoprotein called glycophorin. This is a glycoprotein that is found embedded within the membranes of erythrocytes, more commonly known as your red blood cells. So what we can see here, in addition to one of the stabilizing interactions that we talked about, of course referring to the hydrophobic interactions between the side chains of the transmembrane domain and the fatty acid tails of the phospholipids, we can actually see one other type of stabilizing interaction that helps us out here as well. So for a second, pay attention to what is happening not within the bilayer, but right on top or right beneath it. So if you'll notice, just as the transmembrane domain ends and you start to get into the parts of the protein that are either exposed to the extracellular fluid or exposed to the intracellular fluid, you can see some basic side chains. So on the top side you can see arginine, and on the bottom side you can see a couple of arginines and a couple of lysines. If you'll recall, basic side chains should have a positive electrical charge at a neutral pH value. So what is actually happening here is that these positively charged side chains that are just outside the membrane, they can interact with the negatively charged phosphatidylcholine group that makes up the polar head of the phospholipid of each phospholipid. So what this actually does for us is it holds the transmembrane domain in place. Now, we mentioned before that the phospholipid bilayer is very fluid. It's kind of like when you have a membrane protein, you can kind of imagine kind of like an iceberg floating in the ocean. So you would need a lot of lifting power and a lot of force, but you could pull an iceberg out of the water, or you could possibly have an iceberg sink if you put enough weight on top of it. So we don't want that to happen. We want this transmembrane domain to stay firmly implanted as it is. So having basic side chains right immediately on top and basic side chains right immediately on bottom provides some stabilization so that the transmembrane domain doesn't come slipping out on either side. Way I kind of think of it is it's kind of like a wing nut that you attach onto a screw that's been put into a wall or something. It holds that screw in place and prevents it from over time kind of screwing out or slipping out, as we said. And here's another thing to take note of, and we mentioned this briefly in the previous video. Do you notice how the oligosaccharides, the little sugar chains that are attached to glycophorin, do you notice that they are only on the exterior or outward-facing side of the protein, the part of the protein that faces towards the extracellular fluid? And then notice that you do not see any of these side chains on the side of the protein that faces towards the intracellular fluid. Now, we said in a previous lecture that when it comes to glycosylation of proteins, this is most commonly going to happen at a few different amino acid residues. It can be serine, it can be threonine, it can be asparagine. There are a couple of different amino acids that can act as attachment sites for sugars. So you certainly see a lot of these on the extracellular side, so well, maybe one explanation for why you only see sugars on the extracellular side and not the intracellular side is, okay, well, maybe the intracellular side of the protein just doesn't have those amino acid residues, so maybe we just can't attach sugars there. Well, that is most certainly not the case, because look down here. You can see serines, you can see threonines, you can see asparagines. So we are certainly not lacking for amino acid residues that could theoretically have sugars attached to them. It just doesn't happen. This is what we were trying to emphasize in the previous video. Sugars are only attached to membrane proteins on the extracellular side. It gives the membrane some polarity to it, and it allows the cell, the body, or whatever you want to call it for whatever organism you're talking about, it allows us to tell the difference between the outside part of the membrane and the inside part of the membrane. Very important. Okay, so let's look at another example. So glycophorin was one example in which there was only one transmembrane domain in the integral membrane protein. So that protein, the glycophorin, only goes through the membrane one time. Now, we certainly talked before about examples of proteins that can go through many times. Here's one right here. This is called bacteriorhodopsin. This is a light-sensing protein that is going to be very similar to the G-protein coupled receptor family of proteins that we talked about in the previous video, in that it has 7 transmembrane domains, where each transmembrane domain is its own distinct hydrophobic alpha helix, and you can actually see that illustrated here. And then this is also a special kind of protein because the alpha helices, specifically the side chains that are a part of it, are oriented such that at the center of the protein we can coordinate a molecule of vitamin A, which is more commonly called retinal. Retinal serves as the light-absorbing part of the protein coenzyme complex, and what this allows bacteria to do, or specifically more rarely found archaebacteria that are photosynthetic, this allows them to sense when they're in the presence of light, and then use that as a metabolic stimulus or trigger to start making ATP for themselves. Alright, so that is going to do it for this fairly short video. So this has just been a little bit of an extension of some of the concepts that we talked about at the latter half of the first video. So hope you enjoyed it, and I will see you next time, and we will start talking about beta barrels. See you next time.